Hi everyone, welcome to this M2D2 talk. Our speaker today is Martin Vögele. He's a senior scientist at Schrödinger, and previously he has obtained his PhD in biophysics from the Max Planck Institute of Biophysics. After his PhD, he did a postdoc in computer science at Stanford University, where he worked at the intersection of molecular simulation and machine learning for social biology and drug discovery. Today, he'll be talking about the software package PENSA that he have developed during his postdoc and continue to maintain for the greater good of the community. Thank you so much, Martin, for kind of sharing your work with us here today. And thank you for accepting the invitation as well. Without further ado, the floor is yours.
Thank you so much. Yeah, thanks for having me. I'm going to talk about PENSA and explain a bit what we can do with it, what you can do with it, how it works. I'm happy to take questions in between if they're for clarifying anything I'm explaining. If you want to discuss like broader things, it might be better to keep that for the end of the talk. Okay, so I will first introduce a bit about the problem in molecular simulation, why we need something like an analysis library like PENSA. Then I will mainly explain how we can use it, mostly to compare different ensembles mostly from molecular simulations, but in the future, maybe also more and more from deep generative models that can generate ensembles. So I will first talk about featurization, how we process the features, and then how we can use them to compare that. I will also show some example applications, and in the end, introduce the library itself and tell you where you can find it and what it can do. Okay, the proverbial needle in the haystack is a metaphor that I like very much when I describe what we do in molecular simulations and when we try to analyze them. So what molecular simulations do, for those of you who have not run a classical MD simulation yourself yet, we usually solve Newton's equations of motion, which by themselves are actually pretty simple. However, we have to do these for all the atoms in an entire protein plus solvent, or in DNA and other molecules, co-solvents, whatever. These systems are usually, or can be, very large. Like regular normal size is about 100,000 particles, but people have simulated millions of particles as well. So this can only be done numerically, of course, and on huge supercomputers. Yeah, also another thing that is very complicated here is how these particles interact. We usually simplify these terms a lot, and the simplifications that have worked best so far are, you can see on the right side, we use harmonic bonds to describe all the bonds. So some a bit more complicated functions to describe torsion angles. We use Lennard-Jones interactions for all the non-bonded pairs, and then we explicitly use the electrostatic charges, where we treat each atom as a point charge, another approximation. Okay, so feeding these all together, what we can use this to simulate the temporal evolution of a molecule in space and time. We usually need to start from something, because if we just throw all the atoms in, it will take a long time until they assemble to a three-dimensional form. So we usually need start coordinates that we usually get from an experimental 3D structure, usually from X-ray crystallography or cryo-EM. And the simulations then produce these large ensembles of structures because we model each atom explicitly, they can sample this conformational space in very much detail down to the atomistic scale. So here you can see one of the static structures from the protein data bank. And then in these simulations, you can see how this over time evolves. And you can see some parts are very flexible. For example, this part here shows how this adrenoceptor activates or deactivates over time. However, this is already a simplified representation, so we have to find what is relevant. A very common application is that we can use these simulations not only to simulate what happens in the experiment, but what would happen if. So we want to compare what happens if I put a ligand in the binding pocket at these receptors. What happens if I mutate some amino acids in the sequence of this protein? What happens if I change the pH of the surrounding solvent? And then we simulate 2 or even more conditions, and then we want to compare them. How we usually do this, the simplest way is to just look at these trajectories that are being produced and look at what happens. Then we often define some hand-picked metrics just to quantify what we see, and then calculate that over time with mostly using one-off analysis scripts. And this process can be very, very tedious because, as I have explained, you have 100,000 atoms and you don't know which one is relevant. And in the end, you want something like you can see on the right, where you have like one activation coordinate and a nice graph of which populations of your states shift, and that explains in very simple terms what happens. However, you have all these atoms, and the relevant differences between the 2 conditions, they can be very subtle. Sometimes you have only a shift in minor conformation. So you could, for example, here the major state could not change too much relatively, but like the 2 minor populations change a bit of the probability with which they occur. And this can already have a big effect in the body and in how drugs operate, for example. Also, you have a lot of allosteric effects. Allosteric effects are effects that propagate through a protein, like from one site where, for example, a ligand binds to another site where, for example, they allow binding of another transducer that transmits a signal. And with all this complexity, it's very easy to miss important details. So we don't want to rely on our eyes alone, even if we are trained experts on the systems we study. And for that, people have come up with several systematic approaches of comparing ensembles. And I will highlight 2 of those that have basically inspired PENSA, what we do now. For example, the Anchor library from the Lindorff-Larsen lab, it is a metric for how similar ensembles are. So it calculates basically a single number that will tell you how different one ensemble is from another. This is, for example, very important if you want to see whether your simulation has converged. So you take 2 time intervals in your simulation, and if the ensemble is roughly the same in 2 subsequent long enough time intervals, you can say, okay, my simulation is converged. Or you can compare ensembles that you've created with one force field or another and say, does it make a huge difference if I change that? But it is very difficult to interpret like what's going on under the hood, because most of these metrics are calculated in very abstract spaces, and they don't have too much of an intuitive meaning. Another approach that was used to more specifically find the differences, like show the user where they are DiffNets, and from the Baumann lab. They're basically modified autoencoders, and they are very expressive. So they're very good at finding things. But they, for example, you have to train them on each specific system, and you have to tell it where the mutation site is. So for example, finding differences that stem from a pH change, you would have to adapt that again. And many of the libraries, people write them, they put them online, but then they were designed for a specific problem, and they're very good at solving this specific problem for a specific system. And then you still anyway have to tinker with it yourself. And you have to do this in most cases anyway, our idea was to make a library that basically provides the building blocks to you to build your analysis workflows depending on what you want, but already uses many of these standardized methods that other people have found very useful. So we have kind of find the right balance between standardization and easiness to adapt to your respective problems. And another requirement that we had is that we want to keep it easy and to use and intuitive to interpret. So because we often had these cases that we work with experimental people or with people who are not computational experts, and then you tell them, okay, this is what's going on, but they want to know what exactly they want you to show it in a picture, or they want to know exactly which is the residue or which is the side chain that flips that makes a difference. And so we want to keep it interpretable. So we were thinking of it as a building block approach. And now that we know which approach we want to use, we had to specify which are the building blocks that we want to provide in our library. So what we wanted was flexible featurizations or representations of how we represent the protein to the machine, but also keep it interpretable for us. So we implemented the standard features that many people use for analysis, like torsion angles, like regular bond angles, distances, and so on. But we also keep it flexible by providing an interface for custom featurizations. For example, you can write your own MD analysis function that calculates whatever metric you have in mind that might be relevant to your system. We provide various comparison metrics, like that is similar to Encore, but we want to keep it flexible and also compare various sub-ensembles and the single features if necessary. For those comparison methods, we both want to compare continuous distributions, but also the populations of discrete states. So for each of them, we need slightly different methods. Then also another important thing is what we want to do in a simulation is to find what are the relevant conformations, what are the relevant states, and there we have to distinguish between macro states that describe the system-wide dynamics, like if you have a big change in the conformation of your protein, for example, or microstates, which are local changes, for example, the flip of a side chain. And we also distinguish between those. And also, at all of these stages, we want easy visualization tools where you can visualize either individual residues or get an overview of the overall dynamic of the system. Okay, so here how we decided to put everything together. We basically build up our workflow in 3 stages. The first one is the featurization that uses as an input just the Cartesian coordinates that you get from any kind of your trajectory in your ensemble. The next step is more optional. You can process your feature. You can use dimensionality reduction, collective variables, and the last one is you compare the 2 ensembles. So after finding for that, you can use either direct comparison metrics from statistics, or also we have implemented a novel method that quantifies the information that is shared between two features. Yes, there is a question.
Yes. Like the fact that we have different options to choose from is great, but I was wondering if there are default in the package for, let's say, someone like me who's no expert in MD but have run some MD and want to analyze that. I still need to do some choices and get familiar with the.
Yes, we have some analysis scripts that, for example, will calculate all the torsion angles in a protein and then compare them using a standardized metric. So if you work with a protein you've never seen before, that is usually what I would do. Or if you, we also have an example workflow where you also add the water cavities and then discretize all the microstates and then calculate the state-specific information. I will show later which some examples where we apply them, but we also, we provide those workflows. Basically, we have a standard workflow, but we also keep it optional for everyone to write your own methods. And at each stage, we also provide visualization functions. So we can visualize these features. We can visualize different dimensionality reduction. We can visualize the comparison metrics or the information flow. So when you, for example, when you see in the comparison that there is a certain, like there's a handful of residues that are important, you can then go back and visualize each of them and then start looking at what is relevant, what is going on there. Okay, I have 3 example applications. I will shortly introduce them and then come back to them later once I've explained our workflow. So one of them is the loop opening in a bacterial enzyme. So enzymes usually catalyze chemical reactions, but often they only want to do that in a certain environment. Like in some environment, they just need to pass through, and once they are in the part of the body where they need to exert their function, they want to expose their active site where they catalyze the reaction. And here we have one of those examples. Another one is force field parameters. The parameters at the beginning that I've showed you, they are constantly being updated and improved. For example, for calcium DNA interactions, that has been a long-standing problem, and I will look at how we could have used PENSA to improve these force field parameters. Also, I'm going to show how we trace the signaling pathway through an opioid receptor. Opioid receptors, their function in the cell is to transmit signals from the outside to the inside, and those particular opioid receptors are very important because they work with, as the name already says, with opioids. Are very important in as painkillers, but also have a huge potential for addiction. Okay, so I'm first going to talk about how we're going to tackle those, and then I will come back to those examples. Okay, I said we start with featurization. So we had to think a bit about how we represent a biomolecule best. I will mostly talk about proteins now, but we also provide these functions for DNA and RNA. So first thing that comes through mind is we can use distances. So they are already better than the Cartesian coordinates x, y, z, because if you calculate all the distances between all the atoms, you have a complete representation of your system, and it is independent of the coordinate frame. So if your protein tilts and rotates in space, that doesn't matter. You still get the same description. However, it is a simple choice. However, it is computationally very expensive. Like the more atoms or the more residues you take into account, it grows quadratically. So at some point you will reach the end of the capacities. So if you want to use distance, you should predefine which ones are important to you. For example, just look at the binding site, or just look at some very representative distances, for example, at the ends of these helices. Otherwise, you will just get a picture like this. And this is already, yeah, we already here use a distance cutoff. So even here, not all the distances are shown. So often a better representation will be torsion angles of dihedrals, because luckily, by the chemical nature of the protein backbone bonds, the torsion angles give already a very accurate description of the 3D structure of a protein. So this angle omega here, this is usually rigid, so we don't even have to include it. So we only trace pi and psi usually. This is much more efficient than distances, because if you grow your number of amino acids in your protein chain, this only grows proportionally. And also, like, we usually don't use the raw angles, we use the sine and the cosine. And there are similar angles for DNA and RNA. They are not as simple, so I won't go into them in too much detail, but it's the same principle. Also, we have side chains in proteins. So there we have some additional parameterization. We just use all the rotatable bonds in the side chains, and then we already get most of the variability. And then we trace those angles along these bonds. And with that, we like taking those backbone and side chain torsions together, we already have a pretty complete description of our protein. Another important thing, this one is pretty much novel in PENSA, because not many other analysis codes have that included by default. Water molecules are often very important. That many proteins include small cavities where waters also help to stabilize the protein biohydrogen bonds, or they're even involved in signal transmission, for example, in GPCRs, as we're going to see later. The tricky thing about water molecules is that water molecules are not covalently bound to the protein, so they can exchange. So if you trace one water molecule, but it changes place with another molecule, like for mutation, then the new water molecule will take the position of the old one. So what we usually trace is the water site and not the explicit water molecule itself. We featurize that is a water site either occupied or not, and if it is occupied, we use the dipole moment of the water molecule, like the angle with respect to the z-axis as our feature. We find these cavities via local maxima within a 3D grid. So we align all the structures of the protein to one reference frame. Then we calculate the water density, and then we look for local maxima. And then we have these candidate water sites or water cavities. Okay, so with this complete, hopefully complete description of the protein, we can also build more advanced descriptions from these basic features. You might have heard of dimensionality reduction or clustering, discretization, and so on. So I'm gonna go briefly through the methods that here we have implemented in PENSA. The first one, this is more like for the micro switches, like if you have those rotatable bonds in your side chains, they are on a continuous angle distribution, but usually we know we can describe them in several different states. So they are not kind of rotating, but also most times they flip, like they can make bigger flips, and we're usually more interested in those bigger flips than in the small rotation. So we can discretize these space. And for that, we use multimodal Gaussian fitting. So we fit the distribution of each of these features to a multimodal Gaussian. And then we find the intersects of the Gaussian components of this fitting function and use the intersections as the state boundaries. If we take into account more features in the same discretization, we can also discretize it in multidimensional space. So usually we take together all the side chain angles of one amino acid and use a multidimensional Gaussian discretization. Also, we can use some very basic methods from unsupervised learning, like dimensionality reduction. So we can calculate the principal component in the space of our features, and then we can, for example, get the main components that describe most of the motion that is going on. An important thing here is that if we want to find the differences between two or more ensembles, we have to perform this on the joint data across all ensembles. So we have to pull all the data from our simulations and run the dimensionality reduction on those. That is also something that that in many analysis packages is quite tedious, but we have here written some functions that do that automatically. Once you have these principal components, you can then apply on them the same metrics as for all other continuous features. We also have visualization functions that you can then write out your the frames from your trajectory along this component. So you can then basically, if you put it into a trajectory player like BMD, you can then watch what happens along your component instead of what happens along in the time of the simulation. Okay, once we have these reduced dimensions in this reduced space, we often find that our simulation data clusters together into smaller, clusters of frames, and we want to quantify those. And for that, we have implemented k-means clustering or also regular space clustering. And of course, you can plug in your own more sophisticated clustering methods if necessary. We can also run clustering in raw feature space, but usually we found that doesn't give many good clusters unless you have a small number of features already. Okay, and for those clusters, we can then apply the same metrics as for all other discrete features. I'm going to talk about that in a bit, which is now. Okay, so how can we compare the distributions that we have found in those features and in the processed features? So basically, this mostly boils down to the question: how similar are two distributions? And we compare these two distributions in various ways. The first one is that we can calculate the relative entropy between those two distributions. As we only have a finite number of samples, we discretize them linearly and then calculated the relative entropy for discrete distributions. Another thing one could do, but we haven't implemented, is fitting across fitting analytical functions and then calculating the relative entropy analytically. But yeah, as I said, we chose to do the other way because it's more it's computationally much easier, and it captures more cases. So we discretize this linearly along the feature space and then calculate this relative entropy. However, the relative entropy, as described by the Kullback-Leibler divergence, has some mathematical properties that are not so desirable. For one, it is not symmetrical, so that distance from A to B or from P to Q is different from Q to P. Also, it can diverge numerically if you have empty bins in your discretization. So these two problems are solved by using the Shannon-Jensen divergence, which is basically the same as the Kullback-Leibler, but in a symmetrized version. So you basically calculate it in one direction, calculate it in the other direction, and average it. And this has already very nice mathematical properties. So, and if you take the square root of this divergence, you then get the Shannon-Jensen distance, which is all the nice mathematical properties of a distance metric. One issue here is that the discretization width can affect the results. We have default values that usually work quite well for molecular systems, but be aware that this might occasionally change. We have also implemented a Kolmogorov-Smirnov statistic, which is the maximum difference of the cumulative distributions of two feature distributions. So you have the feature X, you have two distributions. You calculate the cumulative probability across those features and then use the maximum difference. So you do not need any discretization parameter. So you can, for example, also use this to compare it to the Jensen-Shannon divergence. And here for our example, which is a G protein coupled receptor, you can see that for the standard values of the discretization parameter, the results are pretty similar. So you can almost use those interchangeably. Now we have another, a bit more complicated. Oh, yes, there is another question.
No, there's just a few questions in the chat for you.
Yeah.
So the latest one from Alessandra: how do you compare marginal distribution for each angle with discretized GSD, or you compare joint distribution over all the angles?
Sorry, I didn't quite get that. So we compare the distributions. So we compare
Alessandra,
the.
do you mind coming off mute,
yeah.
maybe to clarify the question?
Yeah. So what we do is we have two distributions, and oops, I'm jumping in the simulation. So we have two distributions for the same feature, like one in ensemble A, one in ensemble B. And then we want to know how much do they differ? Like if we have the same distribution, for example, you have a Gaussian distribution around a certain mean with a certain standard deviation. And if you have the same mean and the same standard deviation, both are Gaussian, then they are the same. If, for example, either your mean shifts or your distribution gets broader, in both those cases, the relative entropy will go down. So you have perfect score if both distributions are the same. And for 0, you would go to 0 if they are completely different distributions. That's basically how it works. And we get a similar range of values for the Kolmogorov-Smirnov statistic. I hope that answers the question. I should have probably added a picture of those two distributions.
So, clarifying point, she was curious whether you work with multivariate distribution or with one-dimensional distribution.
In this case, we only work with one-dimensional distributions. So we compare feature by feature. But it's actually a good idea. We could introduce something, but we haven't implemented like multidimensional comparisons yet.
But you are doing that on the PCA result, right? Yes.
For example, you can take into account multiple multiple features, calculate a PCA or any other kind of collective variable, and then compare the distribution along this collective variable. That is possible. But technically, all we can do is a one-dimensional feature comparison. So the multi-dimensional issue is then like just many one-dimensional comparisons.
Okay, cool. Thank you.
Okay, another metric that we use to kind of compare or trace information in a workflow is state-specific information. It is basically the measure for a measure for the mutual information between two features. So basically, kind of similar to a correlation, but it measures the shared information content explicitly, like calculated over this mutual information function. So you can, for example, here in this receptor, we change the protonation state of this residue, and then we can calculate if I did not look at this residue, if I only looked at any of the other residues in which state they are, how certain could I be that from that I correctly guess the state of this aspartate here? That is basically what the state-specific information tells us. Then a further evolution of that is the co-SSI, which measures how much a third variable co-depends on two residues. For example, you have this aspartate, you have another motif down here, and you want to know how much does each water couple the strength of the interaction between those two. So how much does this contribute to the correlation between those two? For example, this one does not do much, but this one does mediates a lot of the shared information between two residues. This requires that we have states as features, so not a continuous features, but states. And those states, of course, they have to be very clearly and very well defined. So that's basically the main reason why we introduced this multimodal Gaussian fitting in the beginning. However, we could also use these metrics on the states we get from clustering methods or any other method that deals with clearly defined proper states. But we mainly use it with those micro switches. Okay, now I promise we're going back to those applications, and we see how this can be useful in practical life for people who do molecular simulations. And our first example was this oxidoreductase, this enzyme, and we wanted to know how does it either protect or open its active site depending on the surroundings. So people found that it might depend on the oxidation state. So they simulated the different oxidation states of this enzyme, and the difference between those two states is mainly in those two cysteine residues, which in the oxidized state form a disulfide bond. So they simulated it once with this disulfide bond and once without. And then they looked at the simulation and finally found a mechanism. And we here tried to show that we could have done it with PENSA much faster, or could have helped them a lot. So what we find with PENSA is like this color distribution. So for each backbone angle and for each side chain angle, we calculate the Jensen-Shannon distance between the oxidized and the reduced state. And then we can immediately see, if we plot this on the original structure, that the differences will be mainly in this region that is, of course, like this region where the cysteines are, that is trivial, but then in the region that is opposed in this cap loop, here, and we correctly identified this phenylalanine to be the main residue involved in the process. So the process is that here this phenylalanine in the oxidized state cannot remain in its original position, but mostly is pushed away a bit by this disulfide bond, and then in the oxidized state can then move closer and further apart, which then opens or closes the gap. Additionally, we also found this hinge region that the original authors could not identify. So we did not just confirm what they found, we also found a bit on top. Okay, our next example were the force field parameters for DNA ion interactions. Here we wanted to know which residues, which force field values reproduce the experimental ensembles best. So we calculated all the DNA backbone torsions and compared them in this time with the Kolmogorov-Smirnov statistic. And our visualization sees that we get this periodic pattern of where the structures do match very badly to the reference ensemble. And the authors of the original paper also saw that. And here you have the deviations in the different force fields. So the second one matches much better. What we provide additionally beyond what they already found is that we can assign single score parameters that quantify how different each force field, how different the results of each force field parameters are from the experimental ensemble. So we can quantify the quality of the parameters, and by that we could enable an automated optimization. So this could basically serve, for example, I heard many of you do machine learning, so you could use it as a loss function, for example, and optimize and then plug in your machine learning model that would optimize the force field parameters. Or in another example, plug in a generative model that is supposed to generate you ensembles of protein structures and then assess how good this model is with a function like this. Like you can either use the average or the maximum, depending on what you want to put your personal focus on. For example, if you want to get rid of those spikes, but overall deviation is fine, or whether you want to completely get rid of any deviations equally. Okay, our last example was the G protein coupled receptor. So I've already mentioned it when talking about the state-specific information. We want to trace the information flow through the receptor. And what we here wanted to know is how important is the presence of the sodium ion for activation or deactivation of the receptor. So activation usually goes on here if this helix moves outwards and makes space for the G protein to bind. So we knew in G protein coupled receptors, water molecules are important. So we included the water cavities in our featurization. And we know that the side chains often work as microswitches. So we include our Gaussian discretization. And in the end, we then calculate the state-specific information for each of those residues. So what we find is the following, that we have indeed this indirect coupling to state changes. Like in these motifs, you don't have to know what these names mean, but we know that these structural motifs are linked to receptor activations. We previously knew that from experiments and from other simulations. Here, of course, the direct coupling in the region proximal to the aspartate is most pronounced, and we found that here it translates this information onto this transmembrane helix 6. So somewhere this interaction must play a very strong role in receptor activation. And indeed, we have confirmed by this that the presence of the sodium ion that influences the protonation state of the aspartate is important for the for receptor activation, and that it indeed works via this aspartate. Additionally, we gained insights into which, on this pathway, which water sites are particularly important, and like for the transmission to this receptor region, especially this water cavity here, we found to be very important, as you can see from the orange color. Okay, so now how can you do all this with the PENSA library? We have put it on GitHub. We have some, we have implemented all the methods described here and a few more. You can browse through the documentation to find what we have done. You can run the example scripts. You can then modify them to run on your own personal system. They're fully compatible with both MD analysis and PyEmma, so you can use those to plug in your own feature readers, your own analysis models like Markov state models and so on. Installation works via pip, and we also have example scripts in the repo. So yeah, check out this repo on GitHub. We've also put a preprint that describes all the example results and describes the rationale behind the software. We put it on the archive. It has the same title as my talk today. So I forgot to put the link here, but you can easily find it by Googling the title of the talk. Yeah, check it out. And in the end, I wanted to thank everyone involved in this project, mainly the PENSA team, my collaborator Neil, who came up with the state-specific information analysis and contributed that to the library. He is a PhD student with Ulrich Zacharia in Dundee, then also my Stanford colleagues, Stang Jasper, and my supervisor, Ron. We got the examples data from Lucas and Sergio, two former colleagues of mine. And of course, we had many beta testers. And we also got funding from Stanford, EMBO, and computation time from Oak Ridge. And yeah, I'll also be super happy if any of you want to try this and use it in your own project. We're always happy about feedback. Just write me an email, open an issue on GitHub, whatever. Think about your own methods and apply it to your projects. Thanks a lot.
Thank you so much, Martin, for sharing your work with us here today. It's quite different from the classical work we have here, people presenting AI method, but at the same time, I really want to have this on because it's quite, I think it's a critical piece in even labeling some of the MD data that people are starting to put out there. People, for example, like the Misato data set recently released 20,000 MD trajectory of protein and ligand, but yet like there's no annotation of what's going on. So like from a machine learning perspective, it's very hard to just use that data. You can use to learn to kind of how to speed up MD or other things, but like to actually know what's happening, if any significant event is quite hard. So very great to have such a library to help us analyze what's happening during these MDs and kind of use that to kind of inform the ML algorithm was kind of useful. So thank you so much again. Any question from the audience? Let me see the chat. Yeah, I think I've missed one question by Madru. I don't know if that person is still here, but the question was, could you explain again the Kolmogorov test again? What's the aim of this test?
Oh, yeah, the Kolmogorov-Smirnov test basically looks for the largest deviation over the, like you integrate over your feature and you look for the largest deviation. So it basically it's another way of telling how different are these distributions. And it puts the focus on the maximum, like on the spot where they diverge the most from each other. It is, I must say, in many cases, practically, it gives very similar results to the Jensen-Shannon divergence, so you can kind of use them interchangeably. They are very cheap to calculate, so I mostly calculate both of them, and then it's also like a good sanity test to see whether they agree with each other.